Okay, so now that we've moved through the Golgi, we want to talk about where we go after the Golgi. So we've gone from the ER to the cis-Golgi, moved from the cis to the medial to the trans-Golgi, and then the trans-Golgi is this massive sorting hub for sending proteins all over the cell. So proteins go from the trans-Golgi to the plasma membrane, proteins go to the lysosome, they go to a whole set of different membrane compartments. But what I want to do is focus on a single transport event, how proteins get from the trans-Golgi to ultimately the lysosome, because this has a slightly different sorting mechanism, and also the lysosome keeps coming back over and over and over again throughout the course because it intersects with multiple different compartments. So understanding how proteins get there and how the control of those enzymatic activities is regulated so that the cell doesn't self-destruct, is how that mechanism is integrated with the sorting mechanism is really important for understanding how the lysosome operates and certain types of diseases such as lysosomal storage diseases. So just as a reminder, what goes on in the lysosome it contains pretty much anything that would break down any macromolecule in the cell. So it contains hydrolytic enzymes, and that includes nucleases for breaking down RNA and DNA, proteases for breaking down proteins, glycosylases for removing sugar groups from proteins, lipases for breaking down lipids, phosphatases for removing phosphates from proteins and other macromolecules, sulfatases for removing sulfate groups from various macromolecules, and then the list goes on and on and on. So pretty much any ACE that you can just call it blank ACE is probably in the lysosome. So almost all of these things, if they were set loose in the cell, would cause the cell to self-destruct. You could destroy the DNA. You could destroy every protein in the cell. You could remove all the sugar groups of things. You could destroy the lipid bilayer. You could wipe out almost all signal transduction. It would be a complete disaster if any of these proteins escape from the lysosome. So, as we mentioned earlier in the class, what has evolved are a series of protective measures. And they're mainly twofold. One, they're contained in the lysosome. So in some sense, they're sent to a very specific prison to keep all those degradative activities trapped in a single location. The other big thing is they're active only at low pH. And you keep a special low pH environment inside the lysosome, so the enzymes are only active there. But if they were somehow to be missorted to end up in a different membrane compartment, or to somehow get outside of the lysosome if it were to rupture, that pH is normally lower than the pH of the other compartments or the cytosol, and as a result, those enzymes would have almost no activity. So it's a two-step containment process. One, it's partitioned into a special compartment, and that compartment has a unique biochemical environment that allows those enzymes to be active there and nowhere else. So what does that look like? Let's just draw it schematically. So if this is the lysosome, what you have in the cytosol is pH around 7.2, but the pH in the lysosome pH is around 5 or 4.5. You can see different numbers in the literature. And what's doing that, what's keeping that low pH, we've already talked about in the context of endocytosis, it's the V-type ATPase. And what that's doing is pumping hydrogen ions into the lysosome and using ATP hydrolysis to do it. So this is primary active transport, pumping hydronium or hydrogen ions into the lysosome, acidifying it, and that's what allows all these enzymes to be active at low pH in the lysosome. So that being said, all of these proteins and also the V-type ATPase all have to be sorted to the lysosome. So what's the sorting mechanism? Obviously, they have to go into the secretory pathway, so they have to have an ER signal sequence. Then they have to move through the Golgi, but once they're in the Golgi, they have to be routed to the appropriate compartment, so they end up in the lysosome. So how is that done? So what I'm going to do is sketch out the Golgi transport to the early lysosome, and then from there it goes on, early endosome, and from there it goes on to the lysosome. So what I'm going to do is start with the trans-Golgi. I'm going to leave a gap for where I'm going to draw a vesicle. So here we are in the trans-Golgi. And let's say I have a protein, my favorite nuclease, that's to be sent to the lysosome. So we need two things, as we've always talked about. You need some sort of address information, and you need some sort of postal service to recognize that zip code and route it to the appropriate place. So in the case of proteins that are supposed to be sorted to the lysosome, the sorting signal is M6P. So what is M6P? M6P is mannose 6-phosphate. So what this is, it's a special glycosylation event that puts a phosphorylated mannose onto the protein. So this is a post-translational modification. So up to this point, every sorting sequence we've seen is actually part of the amino acid sequence. So the signal sequence is a special amino acid sequence. The KDEL sequence is a special amino acid sequence. KKXX is a special amino acid sequence. All of these are baked into the primary sequence of the protein. This is the first one that we've seen where it's a modification to the protein that's read out as a sorting signal. So you have a site that enzymes in the Golgi modify and add this mannose 6-phosphate to. And what happens is you begin to form a vesicle, and I'll talk about how that forms in a second. But what recruits this into the vesicle, so I'm just going to draw this as a dot for mannose 6-phosphate. What, of course, would recognize mannose 6-phosphate except the mannose 6-phosphate receptor? So there's a transmembrane protein that recognizes mannose 6-phosphate on the context of a protein and binds to it and recruits it into this vesicle that's forming at the trans-Golgi. So far we've talked about three things that have coats. We've talked about clathrin in the case of endocytosis. We've talked about COP1 and COP2 vesicle coats in the cases of anterior grade and retrograde transport from the ER to the Golgi. So is this a new type of coat, or is this something we've seen before? So in order to make things maximally confusing, it turns out in this case, the coat is something we've seen before. In this case, it's clathrin. So this is a very interesting case of where the same coat you use for endocytosis also gets used to bud vesicles off of the trans-Golgi. And so there's a whole set of proteins, and we're not going to get into that, that's involved in recruiting clathrin and assembling it onto these vesicles to form a Golgi to lysosome or endosome transport vesicle. So once this vesicle forms, it buds off. We've got the protein with the mannose-6-phosphate on it, and we've got the mannose-6-phosphate receptor bound to that. Of course, then we will get uncoating. I'm not going to draw every step, but what I am going to do is draw it post-fusion. So here we are at the early endosome. And from our discussion of endocytosis, we know that early endosome eventually leads on to the lysosome. So what we've done is we recruited into a vesicle, and it's got clathrin. It eventually ends up intersecting the endocytic pathway and dumping this protein into the early endosome. And depositing the mannose-6-phosphate receptor into the early endosome. So what happens? So why is this a one-way trip? You know, we talked about this with the KDEL receptor, that it only binds on one side of the transport pathway and not on the other. So the KDEL receptor, for example, won't bind KDEL sequences in the ER, but will bind them in the Golgi because of a pH gradient. Here we see something kind of similar. So what we see is that this mannose-6-phosphate gets clipped off in the endosome, and that's because the mannose-6-phosphate, I'll just write it in red, is cleaved at low pH. So there's proteins in the early endosome that are active at the lower pH of the early endosome and that remove this signal. So now this protein's in the endosome and will then traffic through the rest of the endocytic pathway onto the lysosome. And then what we have is a recycling pathway, and we're not going to go into how that works, but to suffice it to say, you recruit the mannose-6-phosphate receptor and you create a vesicle with the receptor, and you send it back. And so the mannose-6-phosphate receptor, just clear this up, is constantly cycling around. It's grabbing mannose-6-phosphate tagged proteins in the TransGolgi, depositing them in the early endosome, but that's a one-way trip because the mannose-6-phosphate gets clipped off. And so why is this an important pathway? Well, one, it's important because it gives us a sense, it's one of the best understood pathways for how proteins get from the Golgi to another compartment. It's also interesting because this pathway intersects with endocytosis, which is why we did that first before doing ER to Golgi trafficking. It's also interesting for a variety of reasons in terms of understanding a set of human genetic diseases called lysosomal storage diseases. So you might imagine, what kinds of diseases would you get if you took out, for example, the enzyme that we were sorting versus taking out, let's say, mannose-6-phosphate receptor, or the enzyme that makes mannose-6-phosphate in the first place. All of these would lead to very different phenotypes. For example, if you took out just one protein that was going to the lysosome, you would cause a very specific defect, and that would lead to one set of symptoms. But if you took out a key component of the sorting machinery, you would prevent all of those proteins from getting to the lysosome, and you would get a much more severe disease. And so this is exactly what we see in patients. So for example, Hurler's disease is a disease that's a defect in glycosaminoglycan. Whereas a disease like eye cell disease or inclusion cell disease they lack the glycnac transferase. So this is part of the enzymatic machinery that adds, or glycnac phosphotransferase, that adds so no addition of M6P. So in one case, what we end up doing is taking out a single enzyme that's destined for the lysosome. So basically, cells can't break down proteins that or macromolecules that have that. That's why it's called a lysosomal storage disease, because what you end up seeing is accumulations of the macromolecules that can't be broken down. But inclusion cell disease is much more severe because you can't sort most, if not all, of the degradative enzymes to the lysosome. So if you take out the machinery, you get a much more severe defect than what you get if you just take out single lysosomal enzymes. Now, why does any of this matter? Because these are severe diseases, and this is always the problem with human genetic diseases, is we can tell you exactly what went wrong, but we often can't do anything about it. And normally I don't cover this in the class, but this is the advantage of screencasting. I can kind of go into this a little bit more. And if you remember, in the transport pathway, in the early endosome, where we ended up was we had our protein that had a mannose-6-phosphate on it that got clipped off, and we had, and I'm just going to draw it as multiple membrane spanning now, the M6P receptor. Now, when I talked about this returning to the TransGolgi. And it certainly does that, and that's probably where most of it goes. But it turns out a fraction of it stays in endosome and then recycles to the plasma membrane. So why is that small fraction that does that matter? So it's interesting from this point of view. So we were talking about this as a closed system, moving from the trans-Golgi to the early endosome, and then back. So it's forming this closed loop. But the early endosome also goes to the lysosome, and then you can also recycle to the plasma membrane. So this is what, for example, if you do endocytosis, the LDL receptor ends up in the endosome and then recycles back to the plasma membrane. So what's happening is there's a small amount of mannose-6-phosphate receptor that traverses this pathway and ends up on the plasma membrane. So who cares? Why would that matter at all? Turns out that this is the absolute key to treating these diseases. And why is that? So let's say you wanted to treat a disease like this. What you'd like to do is consider the cell. You'd like to add the missing enzyme to the outside of the cell, and somehow magically in the lysosome get that protein to go in there, right? Because this protein was absent before, that's what's causing the disease. I would like to inject this and somehow get it to go there. But proteins don't jump across membranes, and they certainly don't jump across two membranes into the cell and then into the lysosome. So how do we get that to happen? So the key to all of this, and I'm just going to pick a random color, is the mannose-6-phosphate receptor. So this small amount of mannose-6-phosphate receptor that ends up on the plasma membrane, if you inject a patient with protein that has mannose-6-phosphate on it, this can then engage with that, trigger endocytosis, and deliver that protein to the lysosome. So this is actually the basis for treating many lysosomal storage diseases. So just by understanding the cell biology, we can actually say, this is what I need to make as a drug. I need to make the protein, and it has to have this modification on it. And if I just inject it into the bloodstream, there's enough mannose-6-phosphate receptor hanging out on the surface that will engage, trigger endocytosis, get pulled in, and intersect and get dumped off in the lysosome, even though this protein isn't coming through the normal pathway. And so this is a really cool understanding of how, if you understand the molecular details of how these pathways work, you can actually design therapies to treat people for whom there's no other way to even think about how to approach their disease. And so this is something that's truly amazing about these lysosomal storage diseases. And I just wanted to point that out since I had the opportunity to spend some more time with that. And so that wraps up this section of the course on ER to Golgi trafficking and trafficking from the Golgi to the lysosome.